review of our complement pathway protease platform. Grant?
Thank you, Nassim. So, when we think about protease therapeutics, one thing comes to mind, and that is proteases are really an ideal therapeutic for high abundancy targets, in particular cascades. For instance, a protease can tackle and inactivate thousands of molecules in its lifetime, allowing for a very efficient regulation of low therapeutic doses. This is in contrast to what we see with antibodies, which require high concentrations in excess of the target, and even higher concentrations when you start looking at small molecules of peptides that also require frequent dosing due to much shorter PK profiles of these types of molecules. At Catalyst, we're developing multiple high-value complement programs. We started out with 2782-PEG, which we partnered to Biogen in late 2019, which is a novel C3 degrader, and it's targeting dry AMD. And we're working with Biogen as a collaboration to move this product forward. Our current lead candidate, CB4332, is leading our spearhead into the complement realm, and I'll talk a little bit more about that today. Following that up is our specific and potent degraders of C4b and C3b. These are the CFI-based proteins that become our ProTune platform, developing CFI-engineered molecules for a wide variety of complement diseases. And following that, we have a new program that we'll be talking about today. It's our Immunotune platform, which is looking at C3a and C5a, immunomodulatory molecules looking at inflammation moving forward. This gives you an overview of where our molecules are playing in a very minimalistic view of the complement cascade. Our C4b and our C3b degraders are targeting the higher ends, upstream initiation of the cascade, either classical lectin pathway or alternative pathway, or being tuned to modulate both pathways with different potencies. We have the center are targeting C3 with a 2782-PEG and CB4332, which is regulating, as Dr. Hainig mentioned, the unmet need of having CFI deficiency and having an unregulated complement cascade when those breaks are just removed from the system. Terminal complement, these are your immune response inflammatory molecules, the aflatoxin, C3a and C5a, and we have new programs targeting these molecules as well. So giving you a quick overview of where we're at with 2782-PEG. This molecule is targeting geographic atrophy, which, as we all know, is a very high market opportunity with a large number of patients. It's been partnered with Biogen in 2019, and we believe this is a best-in-class C3 degrader for dry AMD. It's a protease engineering platform, and we've shown here that with our platform, we can get extended half-life, giving a longer interval in between dosing based upon our non-human primate data. This gives you a closer look at the non-human primate data. If you look at the orange curve on the left-hand panel there, this is the level of C3 in the eye of these non-human primates immediately after giving the C3b 2782-PEG molecule to degrade C3. It is totally degraded before the first time point is taken, and the C3 level stays suppressed for the entire 28 days of the program. On the contrary, we see the PK profile, which is fairly normal for a high extended half-life biologic in the eye, which is going through two levels of decrease in orders of magnitude. So about 90% of the molecules go on, but with that still remaining 10% or less of CB2782, we are maintaining a C3 suppression level. We believe that this data models out to giving us a dosing interval of around 3 or 4 months. This really shows you the power of a protease and how you sweep away difficult to target molecules. If we compare ourselves to the other molecules that are in the clinic right now, targeting dry AMD in the C3 axis, APL2 and NGM621, we are a protease. We can give a much lower dose, as you see, compared to the other molecules, and we believe much less frequently, approximately every 3 or 4 months. We target C3 like the others. However, we have a lot of opportunities in that when we target C3 and we degrade it such that there is no active C3a nor any active C3b. So we cannot form any aflatoxins and it cannot form any convertases. This is in contrast to the other molecules, which are one-to-one binders, which I spoke about before, which are only binding C3b convertases or the C3 molecule to regulate its activity. Stepping on and talking more about 4332. 4332 as our next clinical